The next lecture will be given by Dr. Pavel POLISHCHUK, and he finished his PhD in 2009. In 2015, he established Chemoinformatics lab in Olomouc, in the Czech Republic, and his main interests are in developing method and applying them mainly in discount chemistry. So, Pavel, please, the floor is yours.
Thank you. So today's talk will be about some quite conventional machine learning. So we return back. And I hope that this presentation and this talk will help you to somehow rethink how to apply machine learning for your tasks and revise them. So let's start from the how we know all of us know how is machine learning works. So we have a molecule, we compute some feature vectors, descriptors, embeddings, and then we will searching for the correlation with the biological activity. So straightforward, simple, understandable. However, the question is, for example, is this molecule correct or not? From the chemical perspective, yeah, it seems okay. But is it correct from the biological perspective, this molecule? Because when we model molecules, we should use biologically relevant forms of compounds. Otherwise, we add some noise to our model. In this case, for example, this molecule may exist in two tautomeric states, which differ, and they may differ biological effect, and we do not know a priori which one is more relevant to our biological response. And that's why we cannot solve this issue within the traditional QSR modeling, and we have to choose one of these forms. And probably Alex Tropsha told about this in his opening talk, that we need to standardize those structures. In this case, we just need to choose one tautomer. It can be canonical tautomer or the most stable tautomer predicted by some tools or somewhere else. So we need to choose just one form, but this is not the only complexity we see from this example. There is another dimension. For example, here are different ionized groups which can have different states, protonation states, and this adds another complexity to our molecules. And we also have to consider them, because not all these forms are relevant, are biologically relevant, are really cause a biological response. And again, this dimension is also not the last one. So because molecules inherently are not plain graphs like molecular graphs, but they are 3D because they live in 3D space and they interact in 3D space. And again, we can compute some feature vectors from the 3D structure, applies the machine learning model, some conventional techniques to find the correlation. But molecules do not exist in the one conformer state. Most of them do not exist. And we have multiple of possible conformers. This is already not so clear how to treat this complexity when you have too many forms which can be relevant or may not be relevant, we don't know. And they are represented by multiple feature vectors. And in the traditional QSR, conventional QSR modeling, we do not know how to act with it, how to treat it. Yeah, some solutions were proposed before. In 1997, Hopfinger published his famous paper on 4D QSR, where they tried to treat this complexity of multiple pharmacophores. So it's a very old paper. I will not go through all these steps. So they align all conformers and all molecules all together, then put them into the cubic grid and calculate for every individual molecules the average occupancy by their conformers of these grid cells, and use this information to construct the model. So they try somehow to reduce the complexity in some way to get a single feature vector finally. The issues with this approach was that it depends on the alignment. and it's applicable to quite rigid molecules, because if you have too flexible molecules, this alignment will be reasonable already. It will be too difficult, and if the scaffolds were also these molecules. Some approaches were suggested to apply and use alignment-independent descriptors for 3D molecules. This is one of these examples of such works, where researchers try to model biological activity of imatinib derivatives on ABL kinase. So they took molecules and called them with 2D descriptors, Morgan fingerprints, and obtained pretty good correlation already, 0.7. Then they went to the higher complexity. They docked these molecules with glide, took these poses, calculate for these poses 3D descriptors, and add these descriptors to the previously calculated 2D descriptors, and observed very marginal improvement of. And from every frame, they again compute these feature vectors of 3D descriptors and average them, and add these descriptors to the previous set of descriptors, and again observe very, very, very small improvement. And what is interesting, using these 4D descriptors alone didn't help. So the model was pretty worse. So 0.44 in this case. And all these examples have one joint point. So always researchers tried to reduce complexity to be suitable to apply conventional machine learning models, which can work only on a single feature vector. So they average, most frequently, they average descriptors of individual conformers or instances, and then apply traditional techniques to find the correlation. This is quite good, a working approach, but descriptors, and that's why those 4D models, they were not become popular until now. So they applied in a and we tried to somehow find the workaround, how to solve this issue more naturally. And what was interesting is that we discovered a very old paper, which was published at the same time as the paper of Hopfinger was published, and which was actually solve this issue in a more general way. So it names multi-instance learning, and this paper for the first time formalized this problem. So molecules can be represented by multiple instances, for example, conformers. Active molecules have at least one instance which is biologically relevant, and inactive molecules have all instances which are not biologically relevant. And we have to find somewhere how to separate those actives from inactives using these conditions. The approach which was suggested by Dietrich was following. So they consider molecules as shades. They again align these molecules all together, all conformers all together. Then they emanate 162 rays from the origin in all directions to uniformly represent the space. And then they compute the distance from the origin to the surface for every conformer. And then the algorithm should find the ranges of lower and upper bounds of values, which will mainly match active instances, instances of active molecules, and do not match instances of inactive molecules. Unfortunately, this approach didn't find a lot of applications in chemistry, and actually it was forgotten for a long period of time. Here you can see the list of publications which were published in topic of chemoinformatics on multi-instance learning. So there are just few papers after the Dietrich which were published, and only recently we revisited this topic once more. However, multi-instance learning found nice applications in variety of other fields like speech recognition, video recognition, bankruptcy prediction, many other fields. So it actively developed by the scientific community, but not applying to chemoinformatic tasks. So I hope so. We are in some deceptive stage at this moment yet. So we are still trying to find the way how to apply this, efficiently apply these techniques, and what we can, which benefits we can receive from this multi-instance learning. So here is a scheme which I showed you for the 4D QSAR when we average descriptors to reduce the complexity. And within the multi-instance learning paradigm, it's a so-called back wrapper. So we average descriptors of instances. Another case is instance wrapper. It's also very simple. So we consider every instance, every conformer as individual training sample, and we assign the activity value the same for all instances of the same molecule. Then we again apply single instance algorithm, some conventional random forest, multi-lane perceptron, whatever, compute prediction for every conformer, and then average them to calculate the final prediction. Since we are now in an era of deep learning, neural networks, there were appeared several more approaches which are very similar conceptually to the previous ones. In this case, we get these feature vectors, pass them through the layers of neural network to create some embeddings, and then we average these embeddings and find the correlation with our predicted value. Very similar to the back wrapper. InstanceNet is also quite similar, so we again pass the feature vectors through the neural networks, get some scores, then average them to compute the final prediction. It is not the most sophisticated approach. This one is a more sophisticated one, because in all the previous examples, we treated all instances equally important. So we just average them, and that's all. But actually, not all of these instances are equally relevant. And adding some attention network can somehow help us to solve this issue. So this attention net assign weights to these embeddings of individual instances. And when we calculate the average, we calculate already weighted average. And this architecture has additional feature, which is quite unique, that interpreting these weights, we can identify most contributing instances. In this way, we can identify more relevant or biologically relevant conformers, if we talk about conformers. So we have some ability to interpret these models. That's also quite interesting feature. So now we will switch to some applications where we demonstrate some advantages of this approach. So we took about 200 data sets from Campbell. We calculate up to 100 conformers for every molecule using RDKit with a very large energy window just to discard completely irrelevant conformers and keeping all others. So we do not need to somehow pre-filter those conformers, because we hope that the model will be able to treat all of them, irrespective of the geometry and energy. Then we compute for every conformer 3D pharmacophore descriptors calculated with the PMapper package, which we developed 6 years ago. The idea is very simple. So if we have a pharmacophore, so which represent our conformer, we can split it onto all possible quadruplets. For every quadruplet, we can produce a canonical signature, which encodes labels, distances, and stereochemistry of this quadruplet. And we count the number of identical quadruplets to create the final feature vector. So these feature vectors were very long, so we performed some feature elimination step to reduce the length of these feature vectors. And we apply first different MIL approaches to understand which one is better among those 5 which I mentioned. And interestingly, it was found that instance wrapper was the best one and outperformed all others with a large margin. It's quite interesting. non-intuitive, and currently we do not know why it happens, so we do not have an explanation why it happens, why it works so well. All others were statistically the same, so they are not statistically distinguishable. So they are somewhere on the second place. Okay, so then we compare this instance wrapper and additionally backnet attention is the most comprehensive model with 2D models, which are usually everybody is used for predicting biological activity. And what we found, that instance wrapper even outperform Morgan fingerprints, so models based on the Morgan fingerprints, 2D methods. In many cases, so in 71 cases, it was the best one, and only in 41 cases, Morgan fingerprints perform better than all others. Back attention network was statistically very close and insignificant relatively to the Morgan fingerprints. And other 2D representations like physicochemical descriptors or 2D pharmacophore representation, which is the most closer to the 3D pharmacophore representation we used here, were even worse than these ones. And what we also demonstrated that if we use just a single conformer, we took in this case the best energy conformer in the MMFF force field, we got very bad models, and it's the worst case. So this just shows that we cannot reliably and easily choose the proper conformer for modeling. So we need to consider always conformer ensemble for our modeling. We tried to somehow figure out what is the reasons why on some data sets we observe good results and bad results for 2D, because in some cases, if we compare predictions, these are R-squared tests for different data sets, that in some cases, 2D models performed very poor, while the 3D multi-instance wrapper performed very well. There are some other cases, they are less numerous, where 2D models were better. And it was interesting for us to understand why this difference is observed. We calculate different properties of data sets, trying to create some recommendation maybe for future users. In which case we have to apply multi-instance learning, in which case we can stop on the 2D modeling approach. We found only one statistically significant difference when we compare the fraction of distance scaffolds. So if the fraction of distance scaffolds is lower, then 2D models perform statistically better, more frequently. This is quite easy to explain because in this case, in our data set, we have larger analog series, which is easier to represent by 2D fingerprints. And if we have too many scaffolds which are distinct, they are more suitable for 3D modeling using the multi-instance learning approach. In other cases, we didn't observe statistically significant difference, but we can just conclude that molecules with a lower molecular weight may be more favorable for multi-instance learning, and which have less number of rotatable bonds, so they are more rigid molecules are more suitable for 3D modeling. It's quite common for DQSAR as well. And now is the most exciting part for me, identification of biologically relevant conformers. Can we retrieve from a model, ligand-based model, which doesn't know anything about the structure of a receptor, biologically relevant conformers? And we performed this analysis for 4 sets of compounds, for which these sets were taken from our sets of molecules on which we train our models. All these molecules were in a test set of corresponding data sets. We retrieved for them PDB structures for those which were available in PDB database. We also tried to increase complexity and make the task more challenging. We remove compounds for which RDKit generates conformers very close to native pose. So we selected only molecules for which average RMSD value of their conformers were greater than 2 angstrom. And then we took top 3 conformers predicted by our model, predicted by docking, and compare them with the native pose. And we calculate the RMSD and we count the success case if we observe the difference less than 2 angstrom in these cases. Here you can see this light yellow bar or light orange bars. These are total number of such molecules which satisfy these conditions, which were retrieved from PDB structure. The last bar, which is a dark orange, is the probability to choose randomly correct pose from our conformers. It is pretty high in many cases, and this just indicates that RDKit generates really biologically relevant conformers in multiple cases. So, okay, this is already interesting. What we observed next is that docking performed quite comparable to RDKit, and even a little bit worse in some cases. So, which one is better? It's really questionable. The same we observed if we choose the lowest energy conformer from these molecules. They also result in almost the same performance as docking. And when we choose conformers based on these attention weights, yeah, we again observed comparable performance to other approaches or random choice, but in some cases we observed some improvement over the random selection and all other approaches. And this is, for me, most exciting thing, that we do not know the structure of the receptor, coordinates of the receptor, but we still can find the biologically relevant conformers. It's really interesting. Yeah, another example came from the prediction of a catalytic reaction, actually an enantiomeric excess of chemical reactions. We used 4 data sets of phosphorus catalyst, phase transfer catalyst, and disulfonamide. We encode all these reactions by 2 types of descriptors. So we encode catalyst using, again, we calculate the conformers to represent all these catalysts. For every catalyst, we compute p-mapper descriptor, but in this case, there were not pharmacophore features. We label nodes just by atom element. It is quite easy to customize in p-mapper, and we use only triplets because all these catalysts were used in the same configuration, R or S, so we do not need to use more complex quadruplets because the configuration of all catalysts were identical. And reaction itself was represented by the condensed graph of reaction. So this is a single molecular graph with a dynamic bond, which designate cleaving or forming bonds. And we calculate using SIM tools the fragmental descriptors for these reactions, and we concatenated these feature vectors to get the final training set and pass through the multi-instance learning algorithm to predict delta G value. In some cases, we did not observe improvement over conventional 2D modeling approaches. In some cases, we observed some improvement. Here you can see that this one, we used atom triplets and multi-instance learning. We could perform better than the best 2D approach based on atom pairs. The same happened in the second case. Again, 3D multi-instance learning approach was better than 2D. And what is also important in this case, multi-instance wrapper was also the best one. So these results are for instance wrapper. Again, why it happened, we don't know. That's interesting to explain in future publication and future studies. Another point which we can add, we tried to measure how far we can extrapolate this data. We split the data set on the training example, which is an enantiomeric excess less or equal to the 80%, and all other reactions went to the training, to the test set. And what we observed that 2D models were not able to extrapolate this data set to further and numeric access values, while single instance learning could 3D, using 3D descriptors could at some way, but yeah, correlation is 0, but something better performed, and 3D model also performed a little bit better. Yeah, it's not perfect, but at least it shows that it may benefit in some way, even to extrapolate. Well, in this case, we use this specific quantile loss function to extrapolate better. Here are just other examples. Maybe I will skip them. So these are traditional examples which were used in the past. So we are not only using these neural networks, but there are some other classical approach in multi-instance learning. They exist. And currently, all examples which we talked about belong to the polymorphism case. So we have a molecule and multiple representations. But there are some other cases which are recognized by the multi-instance learning. Part-to-wall, when we consider atoms as instances and molecule as an object. Or segment-to-sequence, when we consider segments as instances and the whole sequence as an object. And I will bring just 2 examples of these cases. These are an example of atoms as instances. There's a quite recent publication. So researchers try to predict macro pKa and micro pKa at the same time. So what they did, they apply graph convolution. So they perform the graph convolution, create atom embeddings. Then they choose only ionizable atoms and make prediction using these embeddings. They predict some scores which can be interpreted as a micro pKa, and those scores further were combined and recalculated into the macro pKa value. So this is very close to the instance net architecture. And what is also interesting is that graph convolution models inherently belongs to the particular cases of multi-instance learning, because we use some pooling function. When we aggregate multiple instances in one single vector, feature vector or embedding, it can be considered as a multi-instance learning solution. And what they observed, so they observed quite high accuracy relatively to the other conventional approaches. And they try to perform interpretation on 3 external test cases of data sets where most acidic atoms were mainly assigned by human experts. And they found that in many cases, there is a good agreement of the prediction of their network with human experts, but not in all cases, of course. So some errors exist. And here is this examples of these errors. So in some cases, it's quite large difference predicted for different centers. But the majority of cases where this model was incorrect belonged to hydroxamic acids, where we have 2 very closely acidic centers, nitrogen and oxygen one. And actually, they're very difficult to distinguish. And all experts assign that the oxygen should be more acidic, but in the solution, there are almost equal distribution of forms is available. So this is a good example how this model can contribute to the solution of such problems. Here is an example when we consider segment as an instance. This is example from already, I would say, bioinformatics field, because in chemoinformatics there are no such examples yet. This major histocompatibility complex class 2 molecules, they're responsible and trigger our immune response. So they recognize the distracted alien proteins in our body and trigger immune response. However, they can bind the sequences from 11 to 30 residues. However, only short subsequence of length 9 is responsible for actual recognition, not the whole sequence. And what they did, they consider with a sliding window, they created these segments, subsequences as instances in a bag, and they try to find the correlation, and they succeeded and found that this approach worked better than other conventional ones. And this is the last example, maybe of some future application. This project, which we recently started in our institution on morphological profiling, the idea is that when we use phenotypic screening in our labs and we identify some good compounds, promising compounds, we need to optimize them further. But to do that, we need to know the target. And in this case, we have to somehow elucidate this target. There are multiple hundreds of tools available online, offline, which can predict the target, but they're not very reliable. So in this case, we wanted to do this in an experimental way. So we perform microscopic images of cells treated with compounds, reference compounds with a known mode of action and unknown mode of action, new compounds. And we made multiple images for the same experiment. We use different dyes to highlight different organelles or part of cells like nucleus, Golgi apparatus, mitochondria, many others. Then these images are segmented and from on individual cells, and from each cell we compute some feature vectors. This could be computed by the conventional techniques, or again by the deep learning, by convolutional networks, some embeddings. But what is done further is the most common step is just average all these vectors obtained from different cells and images all together, concatenate different channels in one, transform to the so-called morphological profile, and use this information for searching of compounds which have similar morphological profiles. If we found such a similar profile within the reference set of compounds, we can hypothesize that our new molecule affects the same biological pathway or even the same target like the reference compound. This is just quite recently started, but we already accumulated a lot of data. Thousands of molecules were screened on multiple cell lines. So we believe that this is something very promising in future. And where the multi-instance learning may contribute here is this part, because we do not, maybe if you will not do simple averaging of these descriptors, we can perform something better. So this is our idea how we can also apply this multi-instance learning problem to something not so, how to say, pure chemoinformatics. But it is still very useful paradigm to think about. So here is our recent review paper, which was published in January. There are two publications regarding the biological activity and anatomic access prediction of different molecules. So if you are interested in this field, you can start reading from our review where we observe all these examples. Here is a set of tools which are available for multi-instance learning. Some of them were quite old. Some of them are not so applicable, easily applicable to chemoinformatic tasks. So we developed our own package, which is freely available on the GitHub. So you can use it, you can try it to apply to your problems. And as a conclusion, I would say that it seems that multi-instance learning is really promising approach. It outperforms in many cases 2D approach, but unfortunately it requires this computation of conformers and feature vectors, and it is more computationally demanding than the 2D models. Therefore, I would suggest you can build 2D model, and if it fails, you can switch to the multi-instance learning model at that point. And maybe you will succeed and you will greatly increase your performance of your models. This feature of identification of key instances is very intriguing for me, and it is still need to discover in future, because there are a lot of approaches how to identify these relevant instances, and it is still underexplored issue. And it is in chemoinformatics, multi-instance learning is like a terra incognita, so there are a lot of white spots around. So we have to answer a lot of questions. I just select just maybe the most major one. What is interesting, just to inspire you, we can think about everything as instances. Instances could be atoms, tautomers, protomeric state, stereoisomers. We can even apply these approaches to model properties of mixtures, because individual components can be considered as instances. And there are specific architectures exist which can use, because in all these cases which I showed you, we consider all instances as independent ones, but exist some architectures which can consider mutual influence of instances in a single bag. So this can be applied to the mixture model. Also, a separate question, how different 3D representation may affect the accuracy of these models. It is still not explored issue. And I would like to thank all collaborators on this project, and the main driving force was Dmitry Zankov, who is now in Japan. So he really contributed a lot to this project and to all these papers and studies. And at the very end, I would like to invite you to our workshop, which we annually organize in Olomouc, in Czech Republic, in winter. The next time it will be in January. It is completely for free, so no fees. It is more oriented on students and young researchers who want to get some practical expertise on different topics. Usually we cover these topics and we split theory and tutorials 50-50. So there are a lot of tutorials during this workshop. So I will be glad to see you there if you decided to attend us. So, and thank you for your attention.
Pardon for an interesting talk, and we have time for a few questions.
Thank you. The talk was very interesting. So when you use like multi-instance 3D approaches, how relevant do you think the ensemble of confirmations you sample is for the performance of the final model?
Repeat please question. How relevant an ensemble of 3D conformers?
Yeah, so how relevant is this? What kind of ensemble I generate? How much depends on that I get a good sample of space, or yeah, how much performance?
We did not specifically investigate this issue, but this can be done in the future studies when we artificially will remove good conformers and look how it performs. Yeah, it's a good suggestion to check.
Okay,
yeah,
thank you.
Question?
I'm interested in the morphological profiling, and you showed the two-dimensional data, but can we restrict ourselves only to the 2D images? I mean, do we need to mind about the three-dimensional topology?
No, in this case, we produced features from images. So we do not add information about molecules, about structures. We can do that, but actually I do not think that this is a good idea, because this can create a large bias in the model.
Yeah.
And in this case, we return to the order AQs with a limited applicability domain and so on. So in this case, we use just 2D images of cells. In theory, it is possible to use 3D cultures and incorporate somehow this imaging of 3D cultures, but we in a very beginning steps.
Okay, thank you.
I have one quick question. So I agree with you that the idea of being able to generate the bioptic conformation from the ensemble is very interesting. And I'm just wondering, if you were to use just simple old-fashioned pharmacophore models to superpose the conformation on ensemble, do you think you'd also be able to do that?
We didn't try, but from my experience, it's extremely difficult when you have a large data set. To create a ligand-based pharmacophore model is very computationally demanding, because you need to align multiple conformers. Yes, there are again some approaches to reduce the complexity when you remove similar molecules to limit the train size of compounds. Actually, it could work, but again, with pharmacophores, there is also an issue that with pharmacophore, you can discover just a single binding mode. In this case, we may hope that if we have molecules having several binding modes, they will be equally well predicted. This is just a hypothesis. So like an ordinary QSAR model, because we do not know binding mode and mechanism of action in some cases when we make this QSAR modeling, but it works, even with mixture of mechanism of action. So then we hypothesize that it should work better than single pharmacophore mode. But again, this should be checked, of course.
Okay, thank you very much.